Hello, everybody. Okay. Hello, everybody. Thank you so much for joining me today. So first, Chris, thanks a ton for hosting and everybody at BITSI. My name is Bernardo Cordoves, and I am the founder and chief science officer of Halo Labs. Today, I'm very excited to talk to you about our core technology and particularly focusing on our latest instrument, the ORCL. The ORCL is a subvisible particle analysis system, and I'll go through what subvisible particle analysis means for those of you who don't do it. But essentially, it's the world's fastest and most intuitive and lowest volume tool that you can use in order to understand the stability, the sterility, and the contamination in your biologic samples. So at the basis of it, what this tool does is that it conducts subvisible particle analysis by imaging, sizing, counting, and actually enabling high throughput identification of the most relevant biological aggregates and particles across the bioprocess. So one of the things that was the most surprising to me as we started inventing this technology and we would go to the different pharma labs is just how ubiquitous subvisible particles and larger aggregates are across essentially all bioformulations. So on the left, you have some large protein aggregates. Those are really big aggregates. They're about 100 microns in equivalent circular diameter. And to me, that was very not coming from this field. It was very counterintuitive just to think that these particles could get so large. But then with these newer modalities, like these gene therapies and AAVs, basically the same pervasive problem exists, which is that formulation development is challenged in that these oftentimes these biologics are very unstable. And even with the more newer modalities like the cell therapies, they can also clump as well. And the nature of these particles can either be the inherent particles, right? So the drug product itself aggregating because it's unstable, or you could have something else like an extrinsic material, as in this figure on the right, you could have something like a fiber embedded into it. And quickly being able to identify these issues, determining whether there's stability or contamination issues is very important, but the tools so far, until we came about, really struggled doing this. Now, so many of you already know this, but subvisible particle analysis, subvisible particles is a critical quality attribute. It's actually the particles are the #1 cause for product recalls. Several companies have actually had to face severe fines and almost shut down because of potentially particulate problems. It is a safety concern, so particles have been tied to immunogenicity and in the worst case, capillary occlusion and anaphylactic shock. But most importantly, that's worst case scenario. But in general, it is an indicator of low short-term stability. A product starts aggregating, and it's essentially lots of particles. Essentially means a poor product. And as a result of that, it is regulated by the FDA, and there's very well-established USP guidelines at the point of lot release. But doing an understanding of the particles in your for your particular system and sample should be had across the bioprocess, not just at the very end. So many of you are probably familiar with different ways to analyze particles. So particles come in all sorts of ranges, right? Some your very small particles, your dimers and trimers in the case of protein aggregates. Those you might measure with something like SEC. As they get a little bit bigger, you may want to use dynamic light scattering. The ones that we're going to be talking about today are greater than 1 micrometer in size. So again, these are very large aggregates, and but they're very important as a result of how much they're linked to immunogenicity. And if the thinnest human capillary can be under 10 micrometers, having particles of that same scale order presents a danger to the patient and a product quality flaw. But one thing that we hear all the time is, hey, I measured my protein aggregate with using SEC. I ran some controlled stress experiments, and I'm seeing that in general, it looks like my formulation is pretty stable. However, once you look at the subvisible particle level, at particles greater than 1 micrometer in size, you could still actually see lots of particles. So just because you don't have small particles, it doesn't mean that you won't have large particles, right? As with all things science, every independent size range has its own characteristics, and they need to be measured. And that was very difficult to do at the earliest of stages until now. So how does this system count in size particles? So it's actually quite simple. So it's a automated membrane microscopy technology. So actually we're using some of the foundations of how subvisible particle analysis was done, but we're bringing it to the 21st century and also adding all of the advanced assays, for example, using fluorescence that enable particle identification. But let's start from the start. So we have an instrument and a consumable. And as you can see, this consumable that we have here is this is a 96 well plate. It's a 96 well filter plate. You can see this metal frame surrounding the edges of these wells. And these black rings are actually hydrophobic in nature. Essentially what that means is when you pipette, it enables 96 independent testing sites over a flat membrane. And this membrane material right here is 400, is composed of polycarbonate with 400 nanometer pores that are very nicely spaced apart. Then basically you load the sample and you take an image, and everything as resulting from the image analysis is automated. Let me walk you through the steps to make this make a little bit more sense. So the first thing that the user would do is you would take this plate that I showed you, put it in the Aura, and the Aura will take an image. So this is an image of a membrane, right? You would see, like all membranes, even though we have really nice optically graded membranes, they will have some texture, right? You might start to see potentially the pores and some lines, and you really don't want to mistake the topography of this texture for the particles that you're trying to measure. So this system will do a pixel by pixel background subtraction to account for the texture of the membrane. And the first step that you do to get that is you just take a picture of the membrane before a sample has been filtered on it. Then you pipette, say 40 microliters or more of your sample of choice in however many wells you want to run. You filter those wells, so just like lemonade, just like making, it's a sieve, right? And the pores are 400 nanometers, and we're imaging particles above 1 micrometer in size. So the pores are much, much smaller than what we're actually imaging. We dry the membrane from the bottom to ensure dry phase imaging, and that's very important because protein aggregates tend to be very translucent, so they tend to disappear with a lot of flow-based techniques. So high contrast is key, and essentially depleting the matrix, the liquid matrix in which the particles are suspended in, is very important. Then we take that plate and then we re-image it. We put it back in the system when we take another picture. So you can see now that you have the topography of the membrane, and superimposed are now the particles that were present, these visible particles that were present in this case in an aggregated protein aggregate sample. So doing image analysis in this would still be very difficult, but because we have the original membrane image, the system's automatically configured to align both of those and get rid of the texture of the background, revealing very small particles in high contrast and enabling high fidelity measurements. And this is really important for biologics because, again, they're very transparent. And imaging biologics in an aqueous background unfortunately results in undercounting and misizing, as has been well published in the literature. But working with this high contrast image, what we do is we start on an image by for every single particle. So we image every particle, and we have the morphological and dimensional details for every single particle. How big it is, in this case, it's 57 microns if you try to turn this particle into a circle, its maximum projection, and even how much it sticks out of plane, because we have built-in dark field sources that enable us to characterize it. So we start with an image for every particle. Then we can also produce probably what is the most popular chart in particle analysis, which is a size distribution. So for every well, how many small particles do we have and how many big particles were present? So typically you want to see very few large particles. And in biologics, this sort of exponential decaying curve is quite common, right? Lots of small particles are present. So we went from a picture for every particle and a characteristic for every particle to a particle size distribution for every well, for every sample. And as we keep going, because this is a high throughput format, it's as long as you basically name the wells with the right with the same conditions, the software will automatically group all of this data and give you high level insight. So in this case, for example, there were 70,000 particles per mil of IgG aggregates greater than 2 microns. This plastic shards made by NIST called ETFE, were measured about 40,000 particles per mil, and the controls held. So we built everything from every single particle to every single particle size distribution across the whole experiment. And it's only 1 minute per sample, and the volumes required are very low. So we can be quantitated down to 5 microliters, as I'll show you in the next few slides. One of the things that this low volume, high throughput approach enables, again, low volumes, 1 minute per measurement per sample, as opposed to tens of minutes, and without even considering cleaning for many of these fluid imaging or light obscuration systems. What we can do now is, for example, do sweep over, measure the stability for different molecules across different buffer and excipient conditions, enable to do high throughput formulation development and actually get the subvisible particle content. And what you can see here is that this BSA was quite unstable. For example, for this B3 citrate buffer, human IgG was for this particular buffer conditions was fairly invariant, but the lysozyme formed lots of particles, and much more so in the condition B12. So this was a 96-well screen, and it took less than 2 hours to process 96 samples and get really tight CVs. You can see those very small error bars showing high fidelity in these measurements. And again, that's enabled because this is a high contrast technology that enables us to resolve very small protein and biological aggregates in high contrast. So as I mentioned before, the system works down to very small volumes. As with this system does not solve statistics, right? In that if there's very few particles, essentially what that means is, hey, it looks like that sample is probably pretty clean and potentially ready to be transferred downstream. It will measure what it's there, and we've shown that down to very low volumes, the results are very consistent. And another thing that when I started this project that wasn't quite intuitive to me is that so this is a filter plate, and it works on a 96-well plate format. So the well diameter is about 5.6 millimeters. And it's amazing how many micrometer scale particles you can fit in a 5.6 millimeter area circle. And here, using 1.6 micrometer beads, we could see that we can get linearity way above 3 million particles per mil. So there's a lot of real estate, and the built-in fluid mechanics of the system are such that if let's say a particle lands on a pore, that pore gets blocked, and the fluid mechanics are such that the streamlines get diverted, and the particles go to the next open spot. So stacking isn't necessarily an issue with the system because it has very advanced membrane technology where the pores are very much separated and they're not essentially intertwined like in a glass fiber membrane. So we talked about imaging, counting, and sizing particles and doing that in very high throughput. But what about ID, right? So it's a very important element, right, which is very information rich. You want to know what these particles are at the very beginning of a potential aggregation point or a failure point so that you don't have to deal with those problems downstream. But that's actually quite challenging. And I'm going to start with a little challenge. These 3 sets of images, top left, top right, and bottom, are from this paper from the Japanese Pharmacopoeia studying NIST's ethylene tetrafluoroethylene. So NIST made these plastic particles that many of you probably have worked with that mimic the morphology, the size distribution of very common protein aggregates, right? So they're low refractive index. It's plastic that basically looks like protein aggregates. And if I were to challenge you, which of these are protein aggregates, just by looking at them, which ones would you guess? And kind of counterintuitively, these 2 sets of images are actually protein aggregates, while the top left third one, including this one that kind of looks like a bunny in the middle, these were actually plastic shards. But I would, if you were to ask me, I would say the top 2 images were probably maybe the plastic aggregates or maybe the bottom, but they look a lot more similar. Now, if you were to use a machine learning model and train these particles based on these attributes, you would basically come to the wrong conclusion. So morphology, and I'm a fan of machine learning, but just relying on morphology alone and just intensity without any additional more sensitive and specific identification mechanism is just simply not enough. Now, morphology is important, but it's no surrogate for particle identification. Now, at the complete opposite end of the spectrum, many of you might probably be professional spectroscopists. You spend a lot of time finding the particle of interest. For example, in this case, this is a Raman signal. You would clean it up, reduce the size of your library to say, hey, what could this possibly be? And then say, hey, I found this 1550 peak. It's probably AMET2. Oh, it's a protein. That takes a lot of time. I love Raman systems. I built Raman systems before, but it's just incredibly time consuming, right? So it's an amazing forensic technology, but to characterize every single particle, it's just simply not built for purpose. If you were to analyze millions of particles, it would probably take a human lifetime. So sensitive as it is, spectroscopy is just not the right solution. What we do is we're leveraging all of the years in fluorescence microscopy and all of the development in using very well-established dyes that have been validated over decades to basically probe the molecular identity or the material identity of the particles of interest. And the system basically uses, it's essentially extending what I showed you before, but now with labeled fluorescence. So the first three steps are the same. The main difference is that we now use these black plates. You might remember that before they looked whitish. We have a black membrane that supports labeled fluorescence workflows because we don't want the membrane to be autofluorescent, right? And then we do the first three steps the same. Take a plate, you background the plate, you apply your sample, and then you measure the particles. And then when you want to ask a question about the particles in nature, you apply a dye of choice, you vacuum them, and then you only measure the fluorescence. So we're not using fluorescence for sizing particles. We're using fluorescence to measure positive fluorescence events, much like in flow cytometry, right? To understand the nature of these particles. So what do the images now look like? So our controls hold. These are alpha blended images. So now your protein aggregates, in this case, they were labeled using thioflavin T, which is one of the most common dyes for studying protein aggregations. It spun off from a lot of Alzheimer's research that's been validated over the years. These blue particles are ETFE, and you can see that they're not red. So red is the fluorescence color. In this case, this blue color actually means that they're not fluorescing. But this SIMI means side illumination membrane imaging. It's essentially our homebrewed dark field. So it means they do not fluoresce. And on top of that, they're sticking at a plane because they're scattering a lot of light when the light comes at an angle. And you can see a mixture of these. So before, and just to drive you back, this is what they look like. They're almost indistinguishable to the human eye. And sorry if I changed this too fast. So this is what the images look like. But now let's get to the data, right? And the particle view software that's in the Aura, it's quite flexible and intuitive. And you can plot all sorts of different parameters against each other. Each one of these dots is a particle. You can't see here, but this is actually the x-axis is just size, and you can change that if you were to want to. The y-axis is the average fluorescence intensity for in this thioflavin T channel. And you can see that there's two discrete populations. So of course, the higher a particle would be in the y-axis, the stronger the fluorescence, right? So we see these weakly fluorescent population. You can have some really big particles, and then this fluorescence cloud. So in this case, the ETFE is plastic. It's not protein. It does not fluoresce. Meanwhile, it positively identified almost 100% of these IgG aggregates using thioflavin T. So it's sensitive and specific. And on top of that, there's very well integrated computation that allows you to break down this protein, non-protein, or whichever question you're trying to answer very quickly. And then on top of that, get into the minutia of every single particle. So I use thioflavin T in the case of protein aggregation to measure protein aggregation and distinguish protein particles from not. Now let's take a look at another common question in protein formulation, and that is polysorbate degradation. So polysorbate is used to stabilize biologics, but it's very well known that now that solutions containing polysorbate 20 and 80, and potentially more so PS20, once they're left even at 4C or at even colder temperatures, over time, the polysorbate can oxidize and form these degraded polysorbate components and particulates. So we use lipid stains to positively identify degraded polysorbate particles, and protein stains to look to get the protein signal. And you can see very easily here how you get two populations. We get our fatty acid population, our protein population, and then a small mixed population. And depending to the degree of mixing, you may get more particles that overlap between the two. And that is just a function of your sample. But this is quite a sensitive assay. We use BODIPI D3821, which is one of the most well-established dyes for labeling lipid particles. We have an application note on the subject, and we have very low limit of detection and quantitation, which is comparable to many of the solution-based assays. But we get to the same sensitivity, plus you have the particles and the images to prove it, so you can see things for with your own very eyes. And the speed is quite great. Again, it's only a minute and a half per sample measured. As we keep going, you say you might have realized. So I discussed using thioflavin T for labeling proteins and BODIPI for labeling lipids. The Aura is a platform that for analyzing subvisible particles, right? So you could use any well-established dye of choice, or basically maybe just leverage billions of years of evolution. So what do I mean by that? Here in this image, you might see some dots here. These are protein aggregates. But after adding trypsin to the top of this membrane, after filtration, you can see how these particles disappear. So we're basically digesting particles using enzymes that are very specific to remove or to attack a population of choice. In this case, we have plastic particles with polysorbate, and then using cell lysis buffer, we can basically degrade lipids, and only the plastic is left behind. So there's more than one question before getting into, for lack of a better word, an intellectual debate about the right dye of choices, or enables you to use your well-established dye and methods of choice to get to the answers. And it's visual and quantitative, right? So let's take this example. We have these human IgG particles, in this case, 35,000 particles per mil, greater than 2 micrometers were counted. We then washed these samples just doing a water rinse. And you can see how we went from 35,000 to 30,000 particles per mil. So about 5,000, 1/7 of that population were soluble particles. And now these are rock solid insoluble protein aggregates. Then we essentially incubated this in trypsin for 5 minutes, basically applying it on the membrane, and you can and then just applying a simple wash, and dramatically, like 90% of the protein population was removed. Again, trypsin only cleaves specific amino acids. Maybe we followed on with chymotrypsin or others, we might have been able to digest the rest. But again, the control cell. So what I'm getting at is that there's many different ways to ask particle questions, and very quickly to characterize an entire population, instead of shooting a Raman laser and trying to make sense of something after 3 hours of signal processing. So let's talk a little bit about volumes, right? So particularly with these gene therapies, low volume analysis is essential. So people already kind of figure out how to scale up biologics with these CHO cells. But with AAV, your HEK cells are non-secreting, producing lots of viruses. It's a little bit more challenging. But you do, and essentially where gene therapy and cell therapy are now is where your protein formulation was maybe 20, 15 years ago. But because these samples are so expensive, low volume analysis is essential. So what can we learn with just say 10 microliters of sample, and to see if in fact AAV is forming these large subvisible particles? So in this case, we have 3 different serotypes, AAVs 2, 5, and 8. We purchased from an AAV vendor. These are unstressed samples, these top 3 images, again, just with 10 microliters of sample. You can see how AAV2 produce very few particles. You don't see lots of particles on the membrane. AAV5, potentially a little bit more, while AAV8, you start seeing much more formation of subvisible particles. And this is before stress, right? Granted, these particles have not been stabilized, no excipients have been used. Then we stress these particles, we submitted them to significant heat stress, so 73 Celsius for 2 hours. And you can see how these particles started to form very large subvisible aggregates. And I may have shown you this image before. We have very discrete large aggregates for AAV2. AAV5 formed these fibrils, so very long tentacle-looking fibril particles, while AAV8 looked more like a denatured smear, if you will. So they responded very differently to these types of stress. So visually, we can already get an understanding of how they're behaving under different stresses. But we also prove to you that, in fact, these are capsid particles aggregating. They're responding to a protein stain, and the other negative controls held. If you don't have a stress sample, you don't measure lots of signal. And we can see these freeze thawing samples, how they start forming a few particles that are present, of course, much less than this very strong thermal stress that we used. This blue line actually means that these particles are actually protruding out of plane. So maybe during aggregation, they form, tightly condensed packed particles. And we can characterize, these three different serotypes in high throughput and low volume for under an array of different conditions. And if we just focus on AAV2, what we can learn is essentially quantitatively is what we saw visually. Clearly, thermal stress produces lots of particles. The control and the rotation and freeze thaw, it didn't look like the rotation and freeze thaw really impacted these particles. They did produce a little bit more than the control, and even a little bit more so in freeze thaw. But the results were quite consistent and with low error bars. Now, here's a very important comment. So we're looking at subvisible particles to get an understanding of the stability of these formulations. Oftentimes, people rely or skip techniques, whether it's from a resources standpoint or from a misunderstanding standpoint. So in this case, we have we cite a very important paper from PharmaRes that came out pretty recently, where they characterized the melting temperatures for different AAV serotypes. And you can see how in AAV2, the melting temperature is 67. AAV5 is supposedly the one that has the highest melting temperature. You would assume that that's potentially the most stable. And AAV8 is right in the middle at 73C. Now let's just focus on AAV2. According to melting to nano DSF, AAV2 at 73C, it would have poor stability, right? It's above the 67C. However, when you actually looked at the particles formed at that temperature, it's the one that formed the least particles. So it's really important to know that different tools measure different things, and they're all important. And things unfolding, particles unfolding versus particles forming large aggregates, they're just phenomenologically different. The good news is that now you can, with these technologies, you can access this visible information and confirm that, in fact, they are AAV aggregates because it's with fluorescent staining. Now, there's more applications, and again, I'm giving you a taste of the different applications of the system. So we're bringing particle analysis to cell line development. So we're working with SUPI samples, complex samples, and are able to understand the stability of, for example, CHO cell lines as early as cell line development. And one way that we do that, that we help with these measurements is, this technique adopts to the most standard fluorescent workflows. So we've adopted antibody labeling. In this case, you can see, we conjugated an antibody that's specifically bound to the human IgG. It didn't stain the lysozyme particles. The lysozyme particles just responded to thioflavin T. So basically, this tool can adapt to your already existing fluorescence assays and antibody assays, sandwich ELISAs, whatever you're using, and give you an understanding from a stability standpoint and a particle formation standpoint, right? So we're not replacing ELISAs, we're just using existing immunohistochemistry techniques to give you an understanding of a particle formation using your, essentially your established assays. Now, what are some of the applications of that? We can distinguish between non-secreting cell lines and secreting cell lines. If we focus on this image in the bottom left, this green signal is the secondary antibody signal showing that, in fact, these CHO cells were producing aggregated antibody at T equals 0. And I think that's very important because more often than not in cell line development, of course, everybody's just interested in finding the best producers, right? It is manufacturing at the end of the day. And they throw down that problem, a little bit downstream to formulation development or pre-formulation. But I think it's very important to know if from the very beginning you're producing junk, right? And I think this is one technology that because you really can't break it, it's not like flow cell that you're going to clog it or anything along those lines. You're going to get a pretty good understanding about whether you're producing very unstable proteins from T equals 0. And again, the different negative controls hold. So what we're seeing is that aggregated antibody or secondary antibody, it is in fact that the drug product is aggregated. So it's easily validatable. Everybody has different ways in which they can perform these workflows. You may have remembered that at one point I said, hey, you can stain the particles of interest after they've been filtered and already measured. Some people want to do more traditional surface chemistry where they incubate it in solution. Both methods work. You get to the same answer either way. Each of these has pros and cons. But the good news is that it's only like a minute per measurement, so you can quickly do the method development of choice should you want to pursue that. But of course, we have already built-in canned solutions that we recommend for you to go with. So last, I'm going to end on cell therapy and basically this new product, the AuraCL, which is designed to study the stability, the viability, and the purity of cell therapy products and cellular products. So why is this important? Cells, as you well know, they're big. They can be 5 or more. Well, bacteria can be even smaller, but they can easily be subvisible. They can be greater than 1 micron pretty much all the time and greater than 10 microns all the time. So for example, trying to pass 788 or something with existing technologies would be very challenging, right? Because the cells are in the same scale order as contaminants potentially. And here's some of the challenges. So, cytometers aren't exactly particle sizers. There's a lot of legacy subvisible particle analysis tools that undercount cell therapy products, and this has very already been well reported in the literature by Lonza. They tend to clog a lot, so they're easy to break. So essentially, flow and a lot of other spin-out techniques, they're really not designed to tackle the stability and the purity for cell therapy products until now. So the Aura leverages essentially solid phase cytometry to do this analysis. And you can use different techniques to inquire the nature of different particles present in a cell therapy. So for example, now in this y-axis, we're plotting a DAPI stain. We're looking for DNA positive signals versus protein positive signals. So we can see that cells, they light up in both. They respond to DNA staining and protein staining. Protein aggregates only respond to protein staining. Non-biological particles do not fluoresce. And we can very quickly get an understanding of what is present in these cell therapy products. And again, because cell therapy is so new and there's potentially billions of cells in a single treatment, they're very difficult to characterize with existing techniques, certainly when it comes to purity. So with this tool, what are some of the information that you can get using AuraCL? So first, you can understand the stability of just the cells themselves. And these are CAR T cells. You can look at, quantify the number of doublets, triplets, quadruplets, or larger aggregates that are present and confirm that in fact they're cellular aggregates because they respond to DNA staining. Next is you can look at foreign contaminants. You may remember this picture from my first slide. So there was a big fiber. It doesn't fluoresce under a DNA stain. And this is an additional large visible particle greater than 100 micrometers in size, something that would probably clog a light obscuration or flow imaging system. And we can, on top of that, confirm what they are. This is an AAV protein aggregate, and in these cases, these are cells embedded inside a fiber. Now, what are some of the key information that you can get from this technique that you really can't from flow cytometry? So flow cytometry is a flow-based technique. It's a really amazing tool. The world's better off for it because of its low contrast, because you're measuring everything in the liquid. Here's an example of what we're seeing. The red line is all of the particles that responded to cellular staining, while the black line is everything else. So we can see that there's a gap between the total population of particles in the cell therapy versus just a cellular population. So there's like a 25% junk here that was basically inaccessible to flow imaging because of the low contrast, or it could also be something else, maybe cell debris. And I'll show you how we can actually quantify that. One way that we do this is using the most established fluorescence fluorescence assays. In this case, you have monocytes. We're looking at monocyte images of monocytes in a membrane. And if we just do some fluorescent, very basic viability staining, if we use a low concentration of a nucleic acid stain, a low concentration of nucleic acid would only permeate dead cells because their cell walls have been compromised. While Chris, as a chemical engineer, will know better than everybody that if you use a high concentration gradient, it's much easier to push things through. So at high concentration, all cellular material will stain. And we can use his differential to do viability assessment. We compare this to existing techniques using flow cytometry and hemocytometry using cell counting chambers and basically the cells that are darkened with in trypan blue are your dead cells. Flow cytometry, we use propylium iodide, but basically for different kinds of cells and different assays, we're basically getting to the same place. So that's really cool. Not necessarily pitching ourselves as a viability replacement tool, but what you can really get with this technology is not only get your cell viability, but get your cell viability in the context of product purity, right? So let's look at this sample on the left, this bar. So it's a cell therapy sample of T cells. And what you see here is that 86.1% of the particles for this unstressed sample were cells, out of which the remaining 14% was something else. It didn't respond to cell staining. Of all of the cell samples, about 82% of them were alive, and about 18 or 19% of those cells were dead. And then there was a number of particles that did not respond to fluorescent staining, but some that did respond to protein staining. Now let's look at a sample that has been heavily degraded. It's 6 rounds of freeze thawing. Same thing. So we got 80% of the samples in this case were cellular. Only 10% were alive. 90% of the cells were dead. But interestingly enough, about most of the remaining sample was actually responsive to protein staining. So potentially several rounds of freeze thaw, it could mean that the HSA, which was used as an excipient in the cell formulation, actually produced protein particles that would have been invisible from flow cytometry. Potentially the cells started shedding some proteinaceous particles. And not only can you get this information, but you can visually verify it in high contrast, right? So you can see particles that are sticking out of plane that respond to all sorts of different stains. So we're really trying to bring in viability in the context of product purity and do it in high throughput. Just to wrap up, there's a couple of other assays that we've shown some really good performance. Oftentimes, I think like the only approved way of expanding T cells right now is using thermodynabeads. I believe that's the case for the 2 approved cell therapy products in the market, or the 2 or the 3. Those are pretty difficult to find using flow cytometry, and other techniques are basically starting to make inroads. But here, very quickly, we can see these dynabeads. These are magnetic beads that are coated with some surface chemistry, that CD3, that basically makes T cells, gives them the signal to start to expand and activate. And we can tell them apart very easily, right? So the dynabeads, they're not cells, so they don't respond to cellular staining or to protein staining, while the cells do. So you can quickly see them in a very untreated complex sample and get quantitative information, right? You can see, we can separate out the dynabeads because they don't fluoresce. They hover about 5 micrometers in size, which is what most of them are spec'd at. And using side illumination, because these are hard particles, they're more rigid, they will scatter more when illuminated at an angle, while cells tend to be a little bit more compliant and they don't. So there's a number of different ways in which we can find these needle in a haystack particles. I think the power of this technology is you can get very high level insights or go down into characterizing every single individual particle. And so I'm very proud of this technology. Wrapping up here, this is really a subvisible particle analysis tools for all biologics, where you get to characterize external impurities, product impurities, stability. It's compatible with USP 788, and as digital imaging, it's in 1788. Enables low sample consumption to high sample consumption. It enables 100% of the particles because, again, everything is in the focal plane in one single surface, not over a channel where you might potentially be missing things out from an out of focus standpoint. It's compatible with protein and cellular assays. And before I keep yapping away, I want to thank Chris and everybody at BITSI one more time for having us. So thank you so much.
All right, thank you, Bernardo. Let's see. There was a question along the way in the chat, which I can go to in a minute. But for those who are online, we do have time. If please pop your questions in the chat, they'll populate up to me, and I can then, if, for example, if there are multiple questions that are sort of the same one, I can compile them into one, or we can just go through them in the order we receive them. All right. One question was, I think, specific to the AAV portion of your presentation. They're thinking particularly on slide 38, and you have the freedom to go back to that if you want to.
Sure. Sorry, I hope I don't make everybody visible.
The AAV concentration, the units that you're using for what was being shown. It may have been one near slide 38.
That one?
Showing. Maybe it's probably this one.
Correct. That's particle. So this is a particle analysis tool. So it's in particles greater than 2 micrometers per mil.
Oh no, I'm misinterpreting the question here. They're trying to understand for the prep when you're making samples. So the AAV samples you were using, how concentrated were they?
I think it's 10 to the. It was straight from the vendor, no dilution, and I think it's either 10 to the 12th or 10 to the 13th genetic copies per unit or GCUs.
Right. Okay. Yep. So they may or may not be all filled capsids, but
correct. That portion of the analysis, we're looking at that application, but that portion of the analysis is not done here.
Okay. So, all right. Good. Thank you, Lauren. We got that question answered for you. Then a different one, when I think more generally being asked is, do all of the samples need to be dried or maybe fully dried before imaging?
So I think that most of these samples at that point in time, they retain some of their hydration, right, while they're being imaged. What we do want to make sure is that much of the liquid matrix is removed so that you can look at these particles independently. So we've heard, oftentimes people, look at that as a potential, hindrance to the measurement, right? Because they want to measure particles in their native state. And essentially it's a trade-off like when you do that. So in the case of protein aggregates, the irreversible aggregates, you're going to capture those, right, with these filtration techniques, no problem. I think what we've shown, and even with these viability measurements, is essentially, it's like, while I support measuring things in the native liquid state, just the power of being able to obtain this high contrast information very quickly is essentially why we exist.
Okay, so I'm going to compile a couple of related questions here. They're asking you to expand a bit. So I'm going to paraphrase here. Can you expand a bit, or maybe recapitulate how this technology is different and or better than tech in the similar sort of size range space or application space, such as MFI?
If you don't mind, I have some slides for that might help explain.
Certainly.
Sorry for the quick passing of the slides. I'm not trying to make anybody dizzy here. So thank you for, I believe they're right here. How do we compare, right? So this is actually some studies that we ran in your lab, Chris, a while ago. We were comparing our tools. So basically, for the most part, right, you would see that we trend the same as other subvisible tools. In general, here's what in light obscuration is notorious for, well, it's the eldest. It's very well established that it's the least sensitive out of all subvisible particle analysis tools, right? So the advantage that there's lots of light obscurity, but a huge disadvantage is that it misses particles, and it's been very well established. For example, in the case of Omontis, right, which was a peptide drug, that it really missed very large subvisible particle aggregates. So we're actually captured using techniques like flow imaging. So we trend the same as them, but we count more. And the reason why we count more is from a refractive index standpoint. So for example, while you might get a particle size distribution in something like, say, MFI that would look like this faint curve, we would probably count more on average, and that's because it's higher refractive index. And the reason for that is that in our case, when you measure these, not in the liquids but in air, you get a much higher contrast, right? So contrast basically means the ability to separate black and white pixels or black and white images. If everything is very faint, it's very difficult to conduct good particle analysis there. You can miss size very easily. And that's actually quite established in papers by Coriolis, where, for example, if you just add a high sucrose concentration, magically your particle counts go down because the liquid's refractive index is going up, right? So again, all techniques, just like in science, right? There's trade-offs in every choices that you make, but refractive index is a very, contrast is a very well-established limitation for light obscuration and flow imaging. So what you get with our technique is you know, the blackers are, pixels are blacker and the whiter pictures are whiter. So if you compare, for example, protein aggregates in BMI, and this is a paper from Coriolis, they will appear much darker and easier to measure, while in MFI, they will be quite translucent, and in FlowCam, well, potentially more discernible than the MFI ones, they're still quite white and potentially prone to, you know, really complicated algorithms to pick all of those up. So we have much higher contrast, right? So you can see why we're typically going to count things more than they are, because the blackers are black. It's easier to conduct better particle analysis in techniques, in images that have higher contrast. So that's where we are.
Okay, so one of you mentioned some of these, but this question that's coming in is a bit more specific, or maybe a bit broader. It's around standards related to, shall we say, comparing results. One of the questions is around verifying. So essentially, you know, I guess, is there a known? And you talked about the NIST ones as well. I think they're actually thinking a bit more different kinds of particles, not just around the ones that NIST chose to focus on.
Yeah, for sure. So we do work with beads as well. I think, you know, beads are so we can confirm sizing using standardized beads. That's a very common tool used. We can also work with count standards. What you see sometimes is like larger beads, you know, beads are not biologics. So during filtration, they may aggregate, right? But one nice thing about the system is that, you know, we have this expression engine. So we can basically just say, hey, what is the total area of particles present in the sample, right? And if you take the total area and divide it by the average diameter of a bead, magically you get the count, right? So sometimes you can get some aggregation with the larger beads, but that's not a problem for us to basically count, you know, to show that we're basically measuring the right area, right? And at the end of the day, all of these techniques, whether it's membrane imaging or flow imaging, you measure your total pixel area and you translate that into a particle and translate that into a size, right? So we can work with well-known count standards and size standards.
Okay, thank you, Bernardo. Let's see. Give it a moment. If anyone has a last question or so you'd like to pop in the chat, please do. Otherwise, I am cognizant of the time, and I realize people will have to log out for their whatever they have probably scheduled for the top of the hour. We have managed to get to all the questions this time. Sometimes we don't manage to get to them, but a lot of them were in a similar direction, so it was easy to compile them. Okay, I'm not seeing any others pop in. Thanks to everyone who was able to join us online. As I said at the beginning, we will convert this recording into something that's downloadable, and I will get that to your BITSE reps that they can share with you as needed, or other colleagues. Bernardo, happy to share a copy with you if you'd like one.
Oh, please, thank you. And thanks again, Chris, for hosting me and everybody for attending.
All right.